Birds. Female left and male right common pheasant, showing that the male is much larger and more colorful than the female. Some bird species, such as this mute swan, do not display sexual dimorphism through their plumage, and instead can be distinguished by other physiological or behavioral characteristics. Generally, male mute swans, or cobs, are taller and larger than females, or pens, and have thicker necks and a more pronounced knob, above their bill. Skeletons of female left and male right black casquid hornbills, Ceratogymna atrata. The difference between the sexes is apparent in the casque on the top of their bill. This pair is on display at the Museum of Osteology. The Eclectus parrot is an example of a bird where the female right is more colorful than the male left. The extinct huia had one of the most bizarre cases of sexual dimorphism in a bird. The larger female right had a long, curved bill adapted to probe into deep holes in living wood, while the smaller male left had a short, chisel-like bill to strip dead wood. Possible mechanisms have been proposed to explain macroevolution of sexual size dimorphism in birds. These include sexual selection, selection for fecundity in females, niche divergence between the sexes, and allometry, but their relative importance is still not fully understood. 66-67 Sexual dimorphism in birds can be manifested in size or plumage differences between the sexes. Sexual size dimorphism varies among taxa, with males typically being larger, though this is not always the case, e.g. birds of prey, hummingbirds, and some species of flightless birds. 68-69 Plumage dimorphism, in the form of ornamentation or coloration, also varies, though males are typically the more ornamented or brightly colored sex. 70 Such differences have been attributed to the unequal reproductive contributions of the sexes. 71 This difference produces a stronger female choice since they have more risk in producing offspring. In some species, the male's contribution to reproduction ends at copulation, while in other species the male becomes the main, or only, caregiver. Plumage polymorphisms have evolved to reflect these differences and other measures of reproductive fitness, such as body condition, 72, or survival. 73 The male phenotype sends signals to females who then choose the fittest, available male. Sexual dimorphism is a product of both genetics and environmental factors. An example of sexual polymorphism determined by environmental conditions exists in the red-backed fairy. Red-backed fairy males can be classified into three categories during breeding season: black breeders, brown breeders, and brown auxiliaries. 72 These differences arise in response to the bird's body condition. If they are healthy they will produce more androgens, thus becoming black breeders, while less healthy birds produce less androgens and become brown auxiliaries. 72 The reproductive success of the male is thus determined by his success during each year's non-breeding season, causing reproductive success to vary with each year's environmental conditions. Migratory patterns and behaviors also influence sexual dimorphisms. This aspect also stems back to size dimorphism in species. It has been shown that the larger males are better at coping with the difficulties of migration and thus are more successful in reproducing when reaching the breeding destination. 74 When viewing this from an evolutionary standpoint, many theories and explanations come into consideration. If these are the result for every migration and breeding season, the expected results should be a shift towards a larger male population through sexual selection. Sexual selection is strong when the factor of environmental selection is also introduced. Environmental selection may support a smaller chick size if those chicks were born in an area that allowed them to grow to a larger size, even though under normal conditions they would not be able to reach this optimal size for migration. When the environment gives advantages and disadvantages of this sort, the strength of selection is weakened and the environmental forces are given greater morphological weight. The sexual dimorphism could also produce a change in timing of migration leading to differences in mating success within the bird population. 75 When the dimorphism produces that large of a variation between the sexes and between the members of the sexes, multiple evolutionary effects can take place. This timing could even lead to a speciation phenomenon if the variation becomes strongly drastic and favorable towards two different outcomes. Sexual dimorphism is maintained by the counteracting pressures of natural selection and sexual selection. For example, sexual dimorphism in coloration increases the vulnerability of bird species to predation by European sparrowhawks in Denmark. 76 Presumably, increased sexual dimorphism means males are brighter and more conspicuous, leading to increased predation. 76 Moreover, the production of more exaggerated ornaments in males may come at the cost of suppressed immune function. 72 So long as the reproductive benefits of the trait due to sexual selection are greater than the costs imposed by natural selection, then the trait will propagate throughout the population. Reproductive benefits arise in the form of a larger number of offspring, while natural selection imposes costs in the form of reduced survival. This means that even if the trait causes males to die earlier, the trait is still beneficial so long as males with the trait produce more offspring than males lacking the trait. This balance keeps dimorphism alive in these species and ensures that the next generation of successful males will also display these traits that are attractive to females. Such differences in form and reproductive benefits arise. The sexual dimorphism could also produce a change in timing of migration leading to differences in mating success within the bird population. 75 When the dimorphism produces that large of a variation between the sexes and the extinct huia. 73 Sexual dimorphism is a product of both genetics and environmental factors. An example of sexual polymorphism determined by environmental conditions exists in the red-backed fairy. Red-backed fairy males can be classified into three categories during breeding season: black breeders, brown breeders, and brown auxiliaries. 72. These differences arise in response to the bird's body condition. If they are healthy, they will produce more androgens, thus becoming black breeders, while less healthy birds produce less androgens and become brown auxiliaries. 72. The reproductive success of the male is thus determined by his success during each year's non-breeding season, causing reproductive success to vary with each year's environmental conditions. Migratory patterns and behaviors also influence sexual dimorphisms. This aspect also stems back to size dimorphism in species. It has been shown that the larger males are better at coping with the difficulties of migration and thus are more successful in reproducing when reaching the breeding destination. 74. When viewing this from an evolutionary standpoint, many theories and explanations come into consideration. If these are the result for every migration and breeding season, the expected results should be a shift towards a larger male population through sexual selection. Sexual selection is strong when the factor of environmental selection is also introduced. Environmental selection may support a smaller chick size if those chicks were born in an area that allowed them to grow to a larger size, even though under normal conditions they would not be able to reach this optimal size for migration. When the environment gives advantages and disadvantages of this sort, the strength of selection is weakened and the environmental forces are given greater morphological weight. The sexual dimorphism could also produce a change in timing of migration, leading to differences in mating success within the bird population. 75. When the dimorphism produces that large of a variation between the sexes and between the members of the sexes, multiple evolutionary effects can take place. This timing could even lead to a speciation phenomenon if the variation becomes strongly drastic and favorable towards two different outcomes. Sexual dimorphism is maintained by the counteracting pressures of natural selection and sexual selection. For example, sexual dimorphism in coloration increases the vulnerability of bird species to predation by European sparrowhawks in Denmark. 76. Presumably, increased sexual dimorphism means males are brighter and more conspicuous, leading to increased predation. 76. Moreover, the production of more exaggerated ornaments in males may come at the cost of suppressed immune function. 72. So long as the reproductive benefits of the trait due to sexual selection are greater than the costs imposed by natural selection, then the trait will propagate throughout the population. Reproductive benefits arise in the form of a larger number of offspring, while natural selection imposes costs in the form of reduced survival. This means that even if the trait causes males to die earlier, the trait is still beneficial so long as males with the trait produce more offspring than males lacking the trait. This balance keeps dimorphism alive in these species and ensures that the next generation of successful males will also display these traits that are attractive to females. Such differences in form and reproductive roles often cause differences in behavior. As previously stated, males and females often have different roles in reproduction. The courtship and mating behavior of males and females are regulated largely by hormones throughout a bird's lifetime. 77. Activational hormones occur during puberty and adulthood and serve to activate certain behaviors when appropriate, such as territoriality during breeding season. 77. Organizational hormones occur only during a critical period early in development, either just before or just after hatching in most birds, and determine patterns of behavior for the rest of the bird's life. 77. Such behavioral differences can cause disproportionate sensitivities to anthropogenic pressures. 78. Females of the Winchat in Switzerland breed in intensely managed grasslands. 78. Earlier harvesting of the grasses during the breeding season lead to more female deaths. 78. Populations of many birds are often male-skewed, and when sexual differences in behavior increase this ratio, populations decline at a more rapid rate. 78. Also, not all male dimorphic traits are due to hormones like testosterone, instead they are a naturally occurring part of development, for example plumage. 79. In addition, the strong hormonal influence on phenotypic differences suggests that the genetic mechanism and genetic basis of these sexually dimorphic traits may involve transcription factors or cofactors rather than regulatory sequences. 80. Sexual dimorphism may also influence differences in parental investment during times of food scarcity. For example, in the blue-footed booby, the female chicks grow faster than the males, resulting in booby parents producing the smaller sex, the males, during times of food shortage. This then results in the maximization of parental lifetime reproductive success. 81. In black-tailed godwits limosa females are also the larger sex, and the growth rates of female chicks are more susceptible to limited environmental conditions. 82. Sexual dimorphism may also only appear during mating season. Some species of birds only show dimorphic traits in seasonal variation. The males of these species will molt into a less bright or less exaggerated color during the off-breeding season. 80. This occurs because the species is more focused on survival than on reproduction, causing a shift into a less ornate state. Consequently, sexual dimorphism has important ramifications for conservation. However, sexual dimorphism is not only found in birds and is thus important to the conservation of many animals. Such differences in form and behavior can lead to sexual segregation, defined as sex differences in space and resource use. 83. Most sexual segregation research has been done on ungulates. 83. But such research extends to bats. 84. Kangaroos. 85. And birds. 86. Sex-specific conservation plans have even been suggested for species with pronounced sexual segregation. 84. The term sesquimorphism, the Latin numeral prefix sesqui means 1 and 1/2, so halfway between mono 1 and d 2, has been proposed for bird species in which both sexes have basically the same plumage pattern, though the female is clearly distinguishable by reason of her paler or washed-out color. 87. 14 examples include Cape sparrow, Passer melanoris. 87. 67. Rufous sparrow, subspecies P. modinensis. 87. 80. And Saxall sparrow, P. amidendri. 87. 245. Non-avian dinosaurs. Examining fossils of non-avian dinosaurs in search of sexually dimorphic characteristics requires the supply of complete and articulated skeletal and tissue remains. As terrestrial organisms, dinosaur carcasses are subject to ecological and geographical influence that inevitably constitutes the degree of preservation. The availability of well-preserved remains is not a probable outcome as a consequence of decomposition and fossilization. Some paleontologists have looked for sexual dimorphism among dinosaurs using statistics in comparison to ecologically or phylogenetically related modern animals. Apatosaurus and Diplodocus. Female Apatosaurus and Diplodocus had interconnected caudal vertebrae that allowed them to keep their tails elevated to aid in copulation. Discovering that this fusion occurred in only 50% of Apatosaurus and Diplodocus skeletons and 25% of Camarasaurus skeletons indicated that this is a sexually dimorphic trait. Theropoda. It has been hypothesized that male theropods possessed a retractable penis, a feature similar to modern-day crocodilians. Crocodilian skeletons were examined to determine whether there is a skeletal component that is distinctive between both sexes, to help provide an insight on the physical disparities between male and female theropods. Findings revealed the caudal chevrons of male crocodiles, used to anchor the penis muscles, were significantly larger than those of females. There have been criticisms of these findings, but it remains a subject of debate among advocates and adversaries. Ornithopoda. Studies of sexual dimorphism in hadrosaurs have generally centered on the distinctive cranial crests, which likely provided a function in sexual display. A biometric study of 36 skulls found sexual dimorphism was exhibited in the crest of 3 species of hadrosaurids. The crests could be categorized as full male or narrow female and may have given some advantage in intersexual mating competition. Ceratopsians. According to Scott D. Sampson, if ceratopsids were to exhibit sexual dimorphism, modern ecological analogs suggest it would be found in display structures, such as horns and frills. No convincing evidence for sexual dimorphism in body size or mating signals is known in ceratopsids, although there is evidence that the more primitive ceratopsian Protoceratops andrewsi possessed sexes that were distinguishable based on frill and nasal prominence size. This is consistent with other known tetrapod groups where mid-sized animals tend to exhibit markedly more sexual dimorphism than larger ones. However, it has been proposed that these differences can be better explained by intraspecific and ontogenic variation rather than sexual dimorphism. 88. In addition, many sexually dimorphic traits that may have existed in ceratopsians include soft tissue variations such as coloration or dewlaps, which would be unlikely to have been preserved in the fossil record. Stegosaurians. A 2015 study on specimens of Hesperosaurus miosi found evidence of sexual dimorphism in the shape of the dermal plates. 2 plate morphs were described, one was short, wide, and oval-shaped, the other taller and narrower. 89-90. Mammals. In a large proportion of mammal species, males are larger than females. Both genes and hormones affect the formation of many animal brains before birth or hatching and also behavior of adult individuals. Hormones significantly affect human brain formation and also brain development at puberty. A 2004 review in Nature Reviews Neuroscience observed that, because it is easier to manipulate hormone levels than the expression of sex chromosome genes, the effects of hormones have been studied much more extensively and are much better understood than the direct actions in the brain of sex chromosome genes. It concluded that while the differentiating effects of gonadal secretions seem to be dominant, the existing body of research support the idea that sex differences in neural expression of X and Y genes significantly contribute to sex differences in brain functions and disease. 91. Pinnipeds. Male and female northern elephant seal, the male being larger with a big proboscis. Marine mammals show some of the greatest sexual size differences of mammals, because of sexual selection and environmental factors like breeding location. 92. The mating system of pinnipeds varies from polygamy to serial monogamy. Pinnipeds are known for early differential growth and maternal investment since the only nutrients for newborn pups is the milk provided by the mother. 93. For example, the males are significantly larger, about 10% heavier and 2% longer, than the females at birth in sea lion pups. 94. The pattern of differential investment can be varied principally prenatally and postnatally. 95. Mironga leonina, the southern elephant seal, is one of the most dimorphic mammals. 96. Primates. Humans. According to Clark Spencer Larson, modern-day Homo sapiens show a range of sexual dimorphism, with average body mass between the sexes differing by roughly 15%. 97. Substantial discussion in academic literature considers potential evolutionary advantages associated with sexual competition, both intrasexual and intersexual, as well as short and long-term sexual strategies. 98. According to Daly and Wilson, the sexes differ more in human beings than in monogamous mammals, but much less than in extremely polygamous mammals. 99. The average basal metabolic rate is about 6% higher in adolescent males than females and increases to about 10% higher after puberty. Females tend to convert more food into fat, while males convert more into muscle and expendable circulating energy reserves. According to Clark Spencer Larson, modern-day Homo sapiens show a range of sexual dimorphism, with average body mass between the sexes differing by roughly 15%. 97. Substantial discussion in academic literature considers potential evolutionary advantages associated with sexual competition, both intrasexual and intersexual, as well as short and long-term sexual strategies. 98. According to Daly and Wilson, the sexes differ more in human beings than in monogamous mammals, but much less than in extremely polygamous mammals. 99. The average basal metabolic rate is about 6% higher in adolescent males than females and increases to about 10% higher after puberty. Females tend to convert more food into fat, while males convert more into muscle and expendable circulating energy reserves. According to Top. Stylized illustration of humans on the Pioneer plaque, showing both male left and female right. Bottom. Comparison between male left and female right pelvises. Tim Hewitt, Director of Research in the Department of Sports Medicine at Ohio State University Wexner Medical Center. Females have, on average, 50-60% of the upper body strength of males, and 80-90% of the lower body strength of males, relative to body size, but females have higher endurance than males. 100. The difference in strength relative to body mass is less pronounced in trained individuals. In Olympic weightlifting, male records vary from 5.5 times body mass in the lowest weight category to 4.2 times in the highest weight category, while female records vary from 4.4 times to 3.8 times, a weight-adjusted difference of only 10-20%, and an absolute difference of about 30%, i.e., 492 kg vs 348 kg for unlimited weight classes. See Olympic weightlifting records. A study, carried out by analyzing annual world rankings from 1980 to 1996, found that males' running times were, on average, 10% faster than females. 101. In early adolescence, females are on average taller than males as females tend to go through puberty earlier, but males, on average, surpass them in height in later adolescence and adulthood. In the United States, adult males are on average 9% taller, 102, and 16.5% heavier, 103, than adult females. Males typically have larger tracheae and branching bronchi, with about 30% greater lung volume per body mass. On average, males have larger hearts, 10% higher red blood cell count, higher hemoglobin, hence greater oxygen carrying capacity. They also have higher circulating clotting factors, vitamin K, prothrombin and platelets. These differences lead to faster healing of wounds and lower sensitivity to nerve pain after injury. 104. In males, pain-causing injury to the peripheral nerve occurs through the microglia, while in females it occurs through the T cells, except in pregnant women, who follow a male pattern. 105. Females typically have more white blood cells, stored and circulating, as well as more granulocytes and B and T lymphocytes. These differences lead to faster healing of wounds and lower sensitivity to nerve pain after injury. 104. In males, pain-causing injury to the peripheral nerve occurs through the microglia, while in females it occurs through the T cells, except in pregnant women, who follow a male pattern. 105. Females typically have more white blood cells, stored and circulating, as well as more granulocytes and B and T lymphocytes. Additionally, they produce more antibodies at a faster rate than males, hence they develop fewer infectious diseases and succumb for shorter periods. 104. Ethologists argue that females, interacting with other females and multiple offspring in social groups, have experienced such traits as a selective advantage. 106, 107, 108, 109, 110. Females have a higher 105. Females typically have more white blood cells, stored and circulating, as well as more granulocytes and B and T lymphocytes. Additionally, they produce more antibodies at a faster rate than males, hence they develop fewer infectious diseases and succumb for shorter periods. 104. Ethologists argue that females, interacting with other females and multiple offspring in social groups, have experienced such traits as a selective advantage. 106, 107, 108, 109, 110. Females have a higher sensitivity to pain due to aforementioned nerve differences that increase the sensation, and females thus require higher levels of pain medication after injury. 105. Hormonal changes in females affect pain sensitivity, and pregnant women have the same sensitivity as males. Acute pain tolerance is also more consistent over a lifetime in females than males, despite these hormonal changes. 111. Despite differences in physical feeling, both sexes have similar psychological tolerance to or ability to cope with and ignore pain. 112. In the human brain, a difference between sexes was observed in the transcription of the PCDH11XY gene pair unique to Homo sapiens. 113. Sexual differentiation in the human brain from the undifferentiated state is triggered by testosterone from the fetal testis. Testosterone is converted to estrogen in the brain through the action of the enzyme aromatase. Testosterone acts on many brain areas, including the SDNPOA, to create the masculinized brain pattern. 114. The brains of pregnant females carrying male fetuses may be shielded from the masculinizing effects of androgen through the action of sex hormone-binding globulin. 115. The relationship between sex differences in the brain and human behavior is a subject of controversy in psychology and society at large. 116. 117. Many females tend to have a higher ratio of gray matter in the left hemisphere of the brain in comparison to males. 118. 119. Males on average have larger brains than females. However, when adjusted for total brain volume, the gray matter differences between sexes are almost non-existent. Thus, the percentage of gray matter appears to be more related to brain size than it is to sex. 120. 121. Differences in brain physiology between sexes do not necessarily relate to differences in intellect. Heyer et al. Found in a 2004 study that men and women apparently achieve similar IQ results with different brain regions, suggesting that there is no singular underlying neuroanatomical structure to general intelligence and that different types of brain designs may manifest equivalent intellectual performance. 122. See the Sex and Intelligence article for more on this subject. Strict graph theoretical analysis of the human brain connections revealed, 123, that in numerous graph theoretical parameters, e.g., minimum bipartition width, edge number, the expander graph property, minimum vertex cover, the structural connectome of women are significantly better connected than the connectome of men. It was shown, 124, that the graph theoretical differences are due to the sex and not to the differences in the cerebral volume, by analyzing the data of 36 females and 36 males, where the brain volume of each man in the group was smaller than the brain volume of each woman in the group. It was shown, 125, that the graph theoretical differences are due to the sex and not to the differences in the cerebral volume, by analyzing the data of 36 females and 36 males, where the brain volume of each man in the group was smaller than the brain volume of each woman in the group. It was shown, 124, that the graph theoretical differences are due to the sex and not to the differences in the cerebral volume, by analyzing the data of 36 females and 36 males, where the brain volume of each man in the group was smaller than the brain volume of each woman in the group. Sexual dimorphism was also described in the gene level and shown to extend from the sex chromosomes. Overall, about 6,500 genes have been found to have sex differential expression in at least one tissue. Many of these genes are not directly associated with reproduction, but rather linked to more general biological features. In addition, it has been shown that genes with sex-specific expression undergo reduced selection efficiency, which leads to higher population frequencies of deleterious mutations and contributes to the prevalence of several human diseases. 125-126. Immune function. Sexual dimorphism in immune function is a common pattern in vertebrates and also in a number of invertebrates. Most often, females are more immunocompetent than males. This trait is not consistent among all animals, but differs depending on taxonomy, with the most female-biased immune systems being found in insects. 127. In mammals, this results in more frequent and severe infections in males and higher rates of autoimmune disorders in females. One potential cause may be differences in gene expression of immune cells between the sexes. 128. Another explanation is that endocrinological differences between the sexes impact the immune system. For example, testosterone acts as an immunosuppressive agent. 129. Cells. Phenotypic differences between sexes are evident even in cultured cells from tissues. 130. For example, female muscle-derived stem cells have a better muscle regeneration efficiency than male ones. 131. There are reports of several metabolic differences between male and female cells. 132. And they also respond to stress differently. 133. Reproductively advantageous. In theory, larger females are favored by competition for mates, especially in polygamous species. Larger females offer an advantage in fertility, since the physiological demands of reproduction are limiting in females. Hence there is a theoretical expectation that females tend to be larger in species that are monogamous. Females are larger in many species of insects, many spiders, many fish, many reptiles, owls, birds of prey and certain mammals such as the spotted hyena, and baleen whales such as blue whale. As an example, in some species, females are sedentary, and so males must search for them. Fritz Vollrath and Jeff Parker argue that this difference in behavior leads to radically different selection pressures on the two sexes, evidently favoring smaller males. 134. Cases where the male is larger than the female have been studied as well. 134. And require alternative explanations. One example of this type of sexual size dimorphism is the bat Myotis nigricans, black Myotis bat, where females are substantially larger than males in terms of body weight, skull measurement, and forearm length. 135. The interaction between the sexes and the energy needed to produce viable offspring makes it favorable for females to be larger in this species. Females bear the energetic cost of producing eggs, which is much greater than the cost of making sperm by the males. The fecundity advantage hypothesis states that a larger female is able to produce more offspring and give them more favorable conditions to ensure their survival. This is true for most ectotherms. A larger female can provide parental care for a longer time while the offspring matures. The gestation and lactation periods are fairly long in M. nigricans, the females suckling their offspring until they reach nearly adult size. 136. They would not be able to fly and catch prey if they did not compensate for the additional mass of the offspring during this time. Smaller male size may be an adaptation to increase maneuverability and agility, allowing males to compete better with females for food and other resources. Female triple-wart sea devil, an anglerfish, with male attached near vent arrow. Some species of anglerfish also display extreme sexual dimorphism. Females are more typical in appearance to other fish, whereas males are tiny rudimentary creatures with stunted digestive systems. A male must find a female and fuse with her. He then lives parasitically, becoming little more than a sperm-producing body in what amounts to an effectively hermaphrodite composite organism. A similar situation is found in the Zeus water bug Phoreticoblia disparata, where the female has a glandular area on her back that can serve to feed a male, which clings to her. Although males can survive away from females, they generally are not free-living. 137. This is taken to the logical extreme in the rhizocephalic crustaceans, like the Saculina, where the male injects itself into the female's body and becomes nothing more than sperm-producing cells, to the point that the superorder used to be mistaken for hermaphroditic. 138. Some plant species also exhibit dimorphism in which the females are significantly larger than the males, such as in the moss Dicranum 139 and the liverwort Sphaerocarpos. 140. There is some evidence that, in these genera, the dimorphism may be tied to a sex chromosome, 140-141, or to chemical signaling from females. 142. Another complicated example of sexual dimorphism is in Vespula squamosa, the southern yellowjacket. In this wasp species, the female workers are the smallest, the male workers are slightly larger, and the female queens are significantly larger than her female workers and male counterparts. Evolution. Sexual dimorphism in Cambrian trilobites 143. In 1871, Charles Darwin advanced the theory of sexual selection, which related sexual dimorphism to sexual selection. 144. The first step towards sexual dimorphism is the size differentiation of sperm and eggs, anisogamy. 145, 146, 147, 148, 917 anisogamy and the usually large number of small male gametes relative to the larger female gametes usually lies in the development of strong sperm competition. 149, 150, because small sperm enable organisms to produce a large number of sperm, and make males or male function of hermaphrodites 151 more redundant. Volvocene algae have been useful in understanding the evolution of sexual dimorphism. 152, and species like the beetle C. maculatus, where the females are larger than the males, are used to study its underlying genetic mechanisms. 153. In many non-monogamous species, the benefit to a male's reproductive fitness of mating with multiple females is large, whereas the benefit to a female's reproductive fitness of mating with multiple males is small or non-existent. 154. In these species, there is a selection pressure for whatever traits enable a male to have more matings. The male may therefore come to have different traits from the female. Male left offspring center and female right Sumatran orangutans. These traits could be ones that allow him to fight off other males for control of territory or a harem, such as large size or weapons. 155. Or they could be traits that females, for whatever reason, prefer in mates. 156. Male-male competition poses no deep theoretical questions. 157. But mate choice does. Females may choose males that appear strong and healthy, thus likely to possess good alleles, and give rise to healthy offspring. 158. In some species, however, females seem to choose males with traits that do not improve offspring survival rates, and even traits that reduce it, potentially leading to traits like the peacock's tail. 157. Two hypotheses for explaining this factor: the sexy son hypothesis and the handicap principle. The sexy son hypothesis states that females may initially choose a trait because it improves the survival of their young, but once this preference has become widespread, females must continue to choose the trait, even if it becomes harmful. Those that do not will have sons that are unattractive to most females, since the preference is widespread and so receive few matings. 159. The handicap principle states that a male who survives despite possessing some sort of handicap thus proves that the rest of his genes are good alleles. If males with bad alleles could not survive the handicap, females may evolve to choose males with this sort of handicap. The trait is acting as a hard-to-fake signal of fitness. Sex differences in intelligence. Sex differences in human intelligence have long been a topic of debate among researchers and scholars. It is now recognized that there are no significant sex differences in average IQ, 1-2, though particular subtypes of intelligence vary somewhat between sexes. 3-4. While some test batteries show slightly greater intelligence in males, others show slightly greater intelligence in females. 3-4. In particular, studies have shown female subjects performing better on tasks related to verbal ability, and males performing better on tasks related to rotation of objects in space, often categorized as spatial ability. 5. Some research indicates that male advantages on some cognitive tests are minimized when controlling for socioeconomic factors. 4. It has also been hypothesized that there is slightly higher variability in male scores in certain areas compared to female scores, leading to males being overrepresented at the top and bottom extremes of the distribution, though the evidence for this hypothesis is inconclusive. 6. IQ research. Background. There is no statistically significant difference between the average IQ scores of men and women. 1.748 average differences have been reported, however, on some tests of mathematics and verbal ability in certain contexts. 4.95 Some studies have suggested that there may be more variability in cognitive ability among males than among females. 9 But others have contradicted this. 10 Or presented evidence that differential variability is culturally rather than biologically determined. 11 According to psychologist Diane Halpern, there are both differences and similarities in the cognitive abilities of women and men, but there is no database rationale to support the idea that either is the smarter or superior sex. 2. Findings. Although most tests show no sex difference, there are some that do. For example, it has been found that female subjects tend to perform better on tests of verbal abilities and processing speed while males tend to perform better on tests of visual spatial ability and crystallized intelligence. 12.13 For verbal fluency, females have been specifically found to perform slightly better, on average, in vocabulary and reading comprehension and significantly higher in speech production and essay writing. 14 Males have been specifically found to perform better, on average, in spatial visualization, spatial perception, and mental rotation. 14 None of these findings, however, suggest an advantage for either sex in general intelligence. 13 Nor in fluid intelligence. 12. Most studies find either a very small difference or no sex difference with regard to general intelligence. 3.15 In 2000, researchers Roberto Colom and Francisco J. Abad conducted a large study of 10,475 adults on 5 IQ tests taken from the primary mental abilities and found negligible or no significant sex differences. The tests conducted were on vocabulary, spatial rotation, verbal fluency and inductive reasoning. 15. The literature on sex differences in intelligence has produced inconsistent results due to the type of testing used, and this has resulted in debate among researchers. 16 Garcia, 2002, argues that there might be a small insignificant sex difference in intelligence in general IQ, but this may not necessarily reflect a sex difference in general intelligence or G factor. 16 Although most researchers distinguish between G and IQ, those that argued for greater male intelligence asserted that IQ and G are synonymous, Lin and Irwin 2004, and so the real division comes from defining IQ in relation to G factor. In 2008, Lin and Irwin proposed that since working memory ability correlates highest with G factor, researchers would have no choice but to accept greater male intelligence if differences on working memory tasks are found. As a result, a neuroimaging study published by Schmidt 2009 conducted an investigation into this proposal by measuring sex differences on an NBAC working memory task. The results found no sex difference in working memory capacity, thus contradicting the position put forward by Lin and Irwin 2008 and more in line with those arguing for no sex differences in intelligence. Sex differences in emotional intelligence. Emotional intelligence A involves using cognitive and emotional abilities to function in interpersonal relationships, social groups as well as manage one's emotional states. It consists of abilities such as social cognition, empathy and also reasoning about the emotions of others. 1. The literature finds women have higher emotional intelligence ability than men based on common ability tests such as the MSCIT. 2 physiological measures and behavioral tests also support this. Finding 3, 4, 5, 6. Emotional intelligence. Emotional intelligence A involves using cognitive and emotional abilities to function in interpersonal relationships, social groups as well as manage one's emotional states. A person with high A ability can perceive, comprehend and express emotion accurately, and also has the ability to access and generate feelings when needed to improve one's self and relationships with others. Women tend to score higher than men on measures of emotional intelligence, but gender stereotypes of men and women can affect how they express emotions. 7. The sex difference is small to moderate, somewhat inconsistent, and is often influenced by the person's motivations or social environment. 7. Basson et al. Say, physiological measures of emotion and studies that track people in their daily lives find no consistent sex differences in the experience of emotion, which suggests that women may amplify certain emotional expressions, or men may suppress them. 7. According to the 4-branch model of emotional intelligence model, there are 4 abilities that exist for emotional intelligence. 8, 9. 1. Perception, the ability to detect and decipher emotions in faces, pictures, voices, and cultural artifact. Also includes the ability to identify one's own emotions. Perceiving emotions represents a basic aspect of emotional intelligence, as it makes all other processing of emotional information possible. 8, 9, 10. 2. Facilitation, the ability to use emotions for various cognitive activities such as thinking and problem solving as well as interacting with others. An emotionally intelligent person can capitalize fully upon his or her changing moods in order to best fit the task at hand. An example of this includes a person using their emotions to motivate themselves. 8, 9. 3. Understanding, the ability to process emotion language and understand why someone, including themselves, might feel a certain way. Understanding emotions also encompasses the ability to be sensitive to slight changes between emotions, and the ability to recognize and describe how emotions evolve over time. 8, 9. 4. Management, the ability to manage one's emotions as well as manage emotional relationship with others. Mate choice. Mate choice is one of the primary mechanisms under which evolution can occur. It is characterized by a selective response by animals to particular stimuli, which can be observed as behavior. 1. In other words, before an animal engages with a potential mate, they first evaluate various aspects of that mate which are indicative of quality, such as the resources or phenotypes they have, and evaluate whether or not those particular traits are somehow beneficial to them. The evaluation will then incur a response of some sort. 1. Mate choice is highly visible in lek mating. Here, black grouse males gather in a quagmire and the females then arrive and observe the male before choosing one. These mechanisms are a part of evolutionary change because they operate in a way that causes the qualities that are desired in a mate to be more frequently passed on to each generation over time. For example, if female peacocks desire mates who have a colorful plumage, then this trait will increase in frequency over time as male peacocks with a colorful plumage will have more reproductive success. 2. Further investigation of this concept has found that it is in fact the specific trait of blue and green color near the eye spot that seems to increase the female's likelihood of mating with a specific peacock. 3. Mate choice is a major component of sexual selection, another being intersexual selection. Ideas on sexual selection were first introduced in 1871 by Charles Darwin, then expanded on by Ronald Fisher in 1915. At present, there are 5 sub-mechanisms that explain how mate choice has evolved over time. These are direct phenotypic benefits, sensory bias, the Fisherian runaway hypothesis, indicator traits and genetic compatibility. In the majority of systems where mate choice exists, one sex tends to be competitive with their same-sex members, and the other sex is choosy, meaning they are selective when it comes to picking individuals to mate with. There are direct and indirect benefits of being a selective individual. 5, 6, 7. In most species, females are the choosy sex which discriminates among competitive males. 4. But there are several examples of reversed roles, see below. It is preferable for an individual to choose a compatible mate of the same species, in order to maintain reproductive success. 8. Other factors that can influence mate choice include pathogen stress and the major histocompatibility complex, MHC. Origins and history. Ronald Fisher in 1913. The peacock tail in flight, the classic example of a Fisherian runaway. Charles Darwin first expressed his ideas on sexual selection and mate choice in his book The Descent of Man, and Selection in Relation to Sex in 1871. He was perplexed by the elaborate ornamentation that males of some species have, because such features appeared to be detrimental to survival and to have negative consequences for reproductive success. Darwin proposed 2 explanations for the existence of such traits. These traits are useful in male-male combat or they are preferred by females. 9. This article focuses on the latter. Darwin treated natural selection and sexual selection as 2 different topics, although in the 1930s biologists defined sexual selection as being a part of natural selection. 10. In 1915, Ronald Fisher wrote a paper on the evolution of female preference and secondary sexual characteristics. 15 years later, he expanded this theory in a book called The Genetical Theory of Natural Selection. There he described a scenario, Fisherian runaway, where feedback between mate preference and a trait results in elaborate characters such as the long tail of the male peacock. In 1948, using Drosophila as a model, Angus John Bateman presented experimental evidence that male reproductive success is limited by the number of mates obtained, while female reproductive success is limited by the number of pregnancies that she can have in her lifetime. 12. Thus a female must be selective when choosing a mate because the quality of her offspring depends on it. Males must fight, in the form of intrasexual competition, for the opportunity to mate because not all males will be chosen by females. This became known as Bateman's principle, and although this was a major finding that added to the work of Darwin and Fisher, it was overlooked until George C. Williams emphasized its importance in the 1960s and 1970s. 13-14. In 1972, soon after Williams's revival of the subject, Robert L. Trivers presented his parental investment theory. Trivers defined parental investment as any investment made by the parent that benefits his or her current offspring at the cost of investment in future offspring. These investments include the costs of producing gametes as well as any other care or efforts that parents provide after birth or hatching. Reformulating Bateman's ideas, Trivers argued that the sex which exhibits less parental investment, not necessarily the male, will have to compete for mating opportunities with the sex that invests more. The differences in levels of parental investment create the condition that favors mating biases. 15. Direct and indirect benefits. The act of being choosy was likely selected for as a way to assess whether or not a potential partner's contributions would be capable of producing and/or maintaining the viability of an offspring. Utilizing these behaviors usually results in two types of benefits to the individual who is being choosy. Direct benefits increase the fitness of the choosy sex through direct material advantages or resources. These benefits include but are not limited to increased territory quality, increased parental care, and protection from predators. There is much support for maintenance of mate choice by direct benefits, 5, and this approach offers the least controversial model to explain discriminate mating. 6. Indirect benefits increase genetic fitness for the offspring, and thereby increase the parent's inclusive fitness. When it appears that the choosy sex does not receive direct benefits from his or her mate, indirect benefits may be the payoff for being selective. These indirect benefits may include high-quality genes for their offspring, known as adaptive indirect benefits, or genes that make their offspring more attractive, known as arbitrary indirect benefits. 7. Inbreeding avoidance. Usually, animal biologists assume that mate choice is biased against relatives because of the negative consequences of inbreeding. 16. However, certain natural constraints act to limit the evolution of inbreeding avoidance, particularly when there is a risk of mating with a partner of a different species, heterospecific mating, and losing fitness through hybridization. 16. Inclusive fitness appears to be maximized in matings of intermediately related individuals. 17. Mechanisms. As of 2018, 5 proposed mechanisms address the evolution of mate choice. Direct phenotypic benefits. Sensory bias. Fisherian runaway. Indicator traits. Genetic compatibility. Direct and/or indirect benefits drive the mating biases described in each mechanism. It is possible that these mechanisms co-occur, although the relative roles of each have not been evaluated adequately. 4. Direct phenotypic benefits. A choosy mate tends to have preferences for certain types of traits, also known as phenotypes, which would benefit them to have in a potential partner. These traits must be reliable, and commutative of something that directly benefits the choosy partner in some way. 18. Having a mating preference is advantageous in this situation because it directly affects reproductive fitness. Direct benefits are widespread and empirical studies provide evidence for this mechanism of evolution. 19-20. One example of a sexually selected trait with direct benefits is the bright plumage of the northern cardinal, a common backyard bird in the eastern United States. Male northern cardinals have conspicuous red feathers, while the females have a more cryptic coloration. In this example, the females are the choosy sex and will use male plumage brightness as a signal when picking a mate. Research suggests that males with brighter plumage feed their young more frequently than males with duller plumage. 21. This increased help in caring for the young lifts some of the burden from the mother so that she can raise more offspring than she could without help. Though this particular mechanism operates on the premise that all phenotypes must communicate something that benefits the choosy mate directly, such selected phenotypes can also have additional indirect benefits for the mother by benefiting the offspring. For example, with the increased help in feeding their young seen in northern cardinals with more plumage brightness, comes an increase in the overall amount of food that is likely to be given to the offspring even if the mother has more children. 22. Though females may choose this trait with the presumed directly advantageous aim of allowing them more time and energy to allocate to producing more offspring, it also benefits the offspring in that 2 parents provide food instead of 1, thereby increasing the likelihood of the overall amount of food available to the offspring despite a possible increase in the amount of offspring siblings. 22. Sensory bias. The sensory bias hypothesis states that the preference for a trait evolves in a non-mating context and is then exploited by the less choosy sex in order to obtain more mating opportunities. The competitive sex evolves traits that exploit a pre-existing bias that the choosy sex already possesses. Following this hypothesis, increased selectivity for one of these specific traits can explain remarkable trait differences in closely related species because it produces a divergence in signaling systems which leads to reproductive isolation. 23. Sensory bias has been demonstrated in guppies, freshwater fish from Trinidad and Tobago. In this mating system, female guppies prefer to mate with males with more orange body coloration. However, outside of a mating context, both sexes prefer animate orange objects, which suggests that preference originally evolved in another context, like foraging. 24. Orange fruits are a rare treat that fall into streams where the guppies live. The ability to find these fruits quickly is an adaptive quality that has evolved outside of a mating context. Sometime after the affinity for orange objects arose, male guppies exploited this preference by incorporating large orange spots to attract females. Another example of sensory exploitation is the case of the water mite Pneumonia papillator, an ambush predator which hunts copepods, small crustaceans passing by in the water column. 25. When hunting, N. papillator adopts a characteristic stance termed the net stance. It holds its first 4 legs out into the water column, with its 4 hind legs resting on aquatic vegetation. This allows it to detect vibrational stimuli produced by swimming prey and to use this to orient towards and clutch at prey. 26. During courtship, males actively search for females. 27. If a male finds a female, he slowly circles around the female whilst trembling his first and second leg near her. 25. 26. Male leg trembling causes females, who are in a net stance, to orient towards and often to clutch the male. 25. This does not damage the male or deter further courtship. The male then deposits spermatophores and begins to vigorously fan and jerk his fourth pair of legs over the spermatophore, generating a current of water that passes over the spermatophores and towards the female. 25. Sperm packet uptake by the female would sometimes follow. 25. Heather Proctor hypothesized that the vibrations made by trembling male legs mimic the vibrations that females detect from swimming prey. This would trigger the female prey detection responses, causing females to orient and then clutch at males, mediating courtship. 25. 28. If this was true and males were exploiting female predation responses, then hungry females should be more receptive to male trembling. Proctor found that unfed captive females did orient and clutch at males significantly more than fed captive females did, consistent with the sensory exploitation hypothesis. 25. Other examples of the sensory bias mechanism include traits in auklets, 29, wolf spiders, 30, and mannequins. 31. Further experimental work is required to reach a fuller understanding of the prevalence and mechanisms of sensory bias. 32. Fisherian runaway and sexy son hypothesis. This creates a positive feedback loop in which a particular trait is desired by a female and present in a male, and that desire for and presence of that particular trait are then reflected in their offspring. 22. If this mechanism is strong enough, it can lead to a type of self-reinforcing coevolution. 22. If runaway selection is strong enough, it may incur significant costs, such as increased visibility to predators and energetic costs to maintain the trait's full expression, hence peacock's extravagant feathers, or any number of mating displays. This model does not predict a genetic benefit, rather, the reward is more mates. In a study done on great reed warblers, models based on the polygyny threshold and sexy son hypotheses predict that females should gain evolutionary advantage in either short-term or long-term in this mating system. Although the importance of female choice was demonstrated, the study did not support the hypotheses. 33. Other studies, such as those conducted on long-tailed widow birds, have demonstrated the existence of female choice. 34. Here, females chose males with long tails, and even preferred those males with experimentally lengthened tails over shortened tails and those of naturally occurring length. Such a process shows how female choice could give rise to exaggerated sexual traits through Fisherian runaway selection. Indicator traits. Indicator traits signal good overall quality of the individual. Traits perceived as attractive must reliably indicate broad genetic quality in order for selection to favor them and for preference to evolve. This is an example of indirect genetic benefits received by the choosy sex, because mating with such individuals will result in high-quality offspring. The indicator traits hypothesis is split into three highly related subtopics: the handicap theory of sexual selection, the good genes hypothesis, and the Hamilton-Zuck hypothesis. People rate the importance of certain traits differently when referring to their own or to others' ideal long-term partners. Research suggests that women consider traits indicating genetic fitness as more important for their own partner, while prioritizing traits that provide benefits to others for their sister's ideal partner. 35. Indicator traits are condition-dependent and have associated costs. Therefore, individuals which can handle these costs well, cf. I can do X here, survive with one hand tied behind my back, should be desired by the choosy sex for their superior genetic quality. This is known as the handicap theory of sexual selection. 36. The good genes hypothesis states that the choosy sex will mate with individuals who possess traits that signify overall genetic quality. In doing so, they gain an evolutionary advantage for their offspring through indirect benefit. The Hamilton-Zuck hypothesis posits that sexual ornaments are indicators of parasite and disessory substance. 37. To test this hypothesis, red jungle fowl males were infected with a parasitic roundworm and monitored for growth and developmental changes. Female preference was also evaluated. The researchers found that parasites affected the development and final appearance of ornamental traits and that females preferred males who were not infected. This supports the idea that parasites are an important factor in sexual selection and mate choice. 38. One of many examples of indicator traits is the condition-dependent patch of red feathers around the face and shoulders of the male house finch. This patch varies in brightness among individuals because the pigments that produce the red color, carotenoids, are limited in the environment. Thus, males who have a high-quality diet will have brighter red plumage. In a much-cited manipulation experiment, female house finches were shown to prefer males with brighter red patches. Also, males with naturally brighter patches proved better fathers and exhibited higher offspring feeding rates than duller males. 20. Genetic compatibility. Genetic compatibility refers to how well the genes of two parents function together in their offspring. Choosing genetically compatible mates could result in optimally fit offspring and notably affect reproductive fitness. However, the genetic compatibility model is limited to specific traits due to complex genetic interactions, e.g. major histocompatibility complex in humans and mice. The choosy sex must know their own genotype as well as the genotypes of potential mates in order to select the appropriate partner. 39. This makes testing components of genetic compatibility difficult and controversial. Role of major histocompatibility complex. A controversial but well-known experiment suggests that human females use body odor as an indicator of genetic compatibility. In this study, males were given a plain t-shirt to sleep in for 2 nights in order to provide a scent sample. College women were then asked to rate odors from several men, some with similar MHC, major histocompatibility complex, genes to their own and others with dissimilar genes. MHC genes code for receptors that identify foreign pathogens in the body so that the immune system may respond and destroy them. Since each different gene in the MHC codes for a different type of receptor, it is expected that females will benefit from mating with males who have more dissimilar MHC genes. This will ensure better resistance to parasites and disease in offspring. Researchers found that women tended to rate the odors higher if the male's genes were more dissimilar to their own. They concluded that the odors are influenced by the MHC and that they have consequences for mate choice in human populations today. 40. Similar to the humans of the odor rating experiment, animals also choose mates based upon genetic compatibility as determined by evaluating the body odor of their potential mates. Some animals, such as mice, assess a mate's genetic compatibility based on their urine odor. 41. In an experiment studying three-spined sticklebacks, researchers found that females prefer to mate with males that share a greater diversity of major histocompatibility complex, MHC, and in addition possess a MHC haplotype specific to fighting the common parasite Gyrodactylus salaris. 42. Mates that have MHC genes different from one another will be superior when reproducing with regard to parasite resistance, body condition and reproductive success and survival. 43. The genetic diversity of animals and life reproductive success, LRS, at the MHC level is optimal at intermediate levels rather than at its maximum, 44-45 despite MHC being one of the most polymorphic genes. 46. In a study, researchers discovered that mice heterozygous at all MHC loci were less resistant than mice homozygous at all loci to Salmonella. It appears disadvantageous to display many different MHC alleles due to the increased loss of T cells, 47, which aid an organism's immune system and trigger its appropriate response. 48. MHC diversity may also correlate with MHC gene expression. As long as a heritable component exists in expression patterns, natural selection is able to act upon the trait. Therefore, gene expression for MHC genes might contribute to the natural selection processes of certain species and be in fact evolutionarily relevant. For example, in another study of three-spined sticklebacks, exposure to parasite species increased MHC class IIB expression by over 25%, proving that parasitic infection increases gene expression. 49. MHC diversity in vertebrates may also be generated by the recombination of alleles on the MHC gene. 50. Sex role reversal in animals. In species where mating biases exist, females are typically the choosy sex because they provide a greater parental investment than males. 51-52. However, there are some examples of sex role reversals where females must compete with each other for mating opportunities with males. Species that exhibit parental care after the birth of their offspring have the potential to overcome the sex differences in parental investment, the amount of energy that each parent contributes per offspring, and lead to a reversal in sex roles. 4. The following are examples of male mate choice, sex role reversal across several taxa. Fish. Male fish typically display high levels of parental care, see pipefish, scissortail sargent, and seahorses. This is because females will deposit their eggs in a special brooding pouch that the male possesses. She does not participate in parental care after this event. The male then has the burden of raising the offspring on his own, which requires energy and time. Thus, males in these species must choose among competitive females for mating opportunities. Surveys across multiple species of pipefish suggest that the sex differences in the level of parental care may not be the only reason for the reversal. Mating systems e.i. monogamy and polygamy might also heavily influence the appearance of male mate choice. 53. Amphibian. Male poison arrow frogs, Dendrobates auratus, take on a very active parenting role. Females are lured by the males to rearing sites where they deposit their eggs. The male fertilizes these eggs and accepts the burden of defending and caring for the young until they are independent. Because the male contributes a higher level of parental investment, females must compete for opportunities to leave their eggs with the limited available males. 54. Bird. Bird species are typically biparental in care, and may also be maternal like the guion and cock of the rocks. However, the reverse may also hold true. Male wattled jacanas provide all parental care after the eggs have been laid by the females. This means that the males must incubate the eggs and defend the nest for an extended period of time. Since males invest much more time and energy into the offspring, females are very competitive for the right to lay their eggs in an established nest. 55. Mammal. There are no confirmed cases of sex role reversed mammals, but female spotted hyenas have peculiar anatomy and behavior that has warranted much attention. 56. Female spotted hyenas are much more aggressive than males due to their high levels of androgens during development. The increased male hormones during development contribute to an enlarged pseudopenis that is involved in mating and birth. 57. Although the anatomical and behavioral roles differ from accepted norms, spotted hyenas are not sex role reversed because the females do not compete with each other for mates. 58. Speciation. For many years it has been suggested that sexual isolation caused by differences in mating behaviors is a precursor for reproductive isolation, lack of gene flow, and consequently speciation, in nature. 59. Mate choice behaviors are thought to be important forces that can result in speciation events because the strength of selection for attractive traits is often very strong. Speciation by this method occurs when a preference for some sexual trait shifts and produces a pre-zygotic barrier, preventing fertilization. These processes have been difficult to test until recently with advances in genetic modeling. 60. Speciation by sexual selection is gaining popularity in the literature with increasing theoretical and empirical studies. There is evidence of early speciation through mate preference in guppies. Guppies are located across several isolated streams in Trinidad and male color patterns differ geographically. Female guppies have no coloration, but their preference for these color patterns also vary across locations. In a mate choice study, female guppies were shown to prefer males with color patterns that are typical of their home stream. 61. This preference could result in reproductive isolation if two populations came into contact again. There is a similar trend shown in two species of the wood-white butterfly, L. reale and L. sinapes. Female L. sinapes controls mate choice by engaging only in conspecific mating, while males attempt to mate with either species. This female mate choice has encouraged speciation of the two wood-whites. 62. The black-throated blue warbler, a North American bird, is another example. Asymmetric recognition of local and non-local songs has been found between two populations of black-throated blue warblers in the United States, one in the northern United States, New Hampshire, and the other in the southern United States, North Carolina. 63 males in the northern population respond strongly to the local male songs but relatively weakly to the non-local songs of southern males. In contrast, southern males respond equally to both local and non-local songs. The fact that northern males exhibit differential recognition indicates that northern females tend not to mate with heterospecific males from the south, thus it is not necessary for the northern males to respond strongly to the song from a southern challenger. A barrier to gene flow exists from south to north as a result of the female choice, which can eventually lead to speciation. Mate choice in humans. In humans, males and females differ in their strategies to acquire mates. Females exhibit more mate choice selectivity than males. According to Bateman's principle, human females display less variance in their lifespan reproductive success, due to their high obligatory parental investment. 64. Human female sexual selection is indicated by sexually dimorphism, especially in traits that serve little other evolutionary purpose, such as the presence in men of beards, overall lower voice pitch, and average greater height. Women have reported a preference for men with beards and lower voices. 65-66 The traits most salient to female human mate choice are parental investment, resource provision and the provision of good genes to offspring. 67 Women as well as men may seek short-term mating partners. 68 This could gain them resources, provide genetic benefit, as through the sexy son hypothesis, facilitate a desired breakup, and allow them to assess a mate's suitability as a long-term partner. 67 Women prefer long-term partners over short-term mates, as they have a larger investment in a child through pregnancy and lactation. 67 Factors in female mate choice include the woman's own perceived attractiveness, the woman's personal resources, mate copying and parasite stress. 67 Romantic love is a mechanism through which long-term mate choice occurs in human females. 69 In humans, females have to endure a 9-month pregnancy and childbirth. 67 Females thus provide a greater biologically obligatory parental investment to offspring than males. 67-70 This provides males with a greater window of opportunity to mate and reproduce than females, hence females are usually more choosy, but males still make mate choices. 70 When finding a short-term mate, males highly value women with sexual experience and physical attractiveness. Men seeking short-term sexual relationships are likely to avoid women who are interested in commitment or require investment. 71 For a long-term relationship, males may look for commitment, facial symmetry, femininity, physical beauty, waist-hip ratio, large breasts, 72 and youth. 73, 69, 74, 75, 76 Due to the higher obligatory biological investment, women are choosier in short-term mating, as the perceived paternal investment is low to non-existent, whereas men and women are equally choosy when deciding for long-term mates, as men and women then have an equal parental investment, as men then invest heavily in the offspring by resource provisioning. 77. The parasite stress theory suggests that parasites are diseases stress an organism, making them look less sexually attractive. 78 Choosing a mate for attractiveness could thus help to find a healthy mate resistant to parasites. 79, 80 Scarification could be viewed by prospective mates as evidence that a person has overcome parasites and is thus more attractive. 81, 82 Masculinity, especially in the face, could equally indicate robust parasite-free health. 83, 84, 85, 86 Polygamy is predicted by pathogen stress in the tropics. 87, 88 Human leukocyte antigen HLA proteins are essential for immune system functioning and are highly variable, assumed to be a result of frequency-dependent parasite-driven selection and mate choice. There is some evidence that women detect and select HLA type by odor, though this is disputed. 89, 90, 91, 92, 93 Human facial preferences correlate with both MHC similarity and MH heterozygosity. 94. Mate choice for cognitive traits. In the late 19th century, Charles Darwin proposed that cognition, or intelligence, was the product of two combined evolutionary forces, natural selection and sexual selection. 95 Research on human mate choice showed that intelligence is sexually selected for, and is highly esteemed by both sexes. 96-97 Some evolutionary psychologists have suggested that humans evolved large brains because the cognitive abilities associated with this size increase were successful in attracting mates, consequently increasing reproductive success. Brains are metabolically costly to produce and are an honest signal of mate quality. 98 Cognition may be functioning to attract mates in other taxa. 99 If the possession of higher cognitive skills enhances a male's ability to gather resources, then females may benefit directly from choosing more intelligent males, through courtship feeding or all-o feeding. 100 Assuming cognitive skills are heritable to some degree, females may also benefit indirectly through their offspring. 99 Additionally, cognitive ability has been shown to vary significantly, both within and between species, and could be under sexual selection as a result. 101 Recently, researchers have started to ask to what extent individuals assess the cognitive abilities of the opposite sex when choosing a mate. 99 In fruit flies, the absence of sexual selection was accompanied by a decline in male cognitive performance. 102 In non-human vertebrates, female preference for males with enhanced cognitive ability may be reflected in successful males' courtship displays, foraging performance, courtship feeding or diet-dependent morphological traits. 99 However, few are the studies that assess whether females can discriminate between males through direct observation of cognitively demanding tasks. Instead, researchers generally investigate female choice by reason of morphological traits correlated with cognitive ability. 99 A male satin bowerbird guards its bower from rival males in the hopes of attracting females with its decorations. Birds. Budgerigars. In this parrot species, direct observation of problem-solving skills was shown to increase male attractiveness. 103. In a two-step experiment testing for female preference, initially less preferred male budgerigars became preferred after females observed them solve extractive foraging problems while initially preferred males failed to problem solve. This shift in preference suggests that females discriminate between males through direct observation of cognitively demanding tasks. 103. Zebra finches. Researchers conducted a problem-solving experiment similar to the one described above. 104. However, male problem-solving performance was not found to influence female mating preferences. Instead, females had a significant preference for males with better foraging efficiency. There is no evidence that females indirectly assess male cognitive ability through morphological traits. By selecting for males with a higher foraging efficiency, females obtain direct food benefits. 104. Satin bowerbirds. Bowerbirds are famous because of their complex male courtship, in which they utilize decorations of certain colors. 105. This suggests that male bowerbirds with enhanced cognitive abilities should have a selective advantage during male courtship. 106. In a study testing problem-solving skills, male satin bowerbirds' general cognitive performance was found to correlate to mating success, rendering them more sexually attractive. 106. However, a more recent study evaluated bowerbirds on 6 cognitive tasks, and found very little intercorrelation between them. 107. In a more recent study, bowerbird performance in 6 cognitive tasks was positively correlated with mating success, but no general cognitive ability was found as intercorrelations between performances on each task were weak. Instead, mating success could be independently predicted via the male's performance on most tasks. This demonstrates the complexity of the link between cognitive ability and sexual selection. 107. Red crossbills. An experimental study showed that female red crossbills prefer males with higher foraging efficiency. 108. After observing 2 males simultaneously extracting seeds from cones, females spent more time in proximity of the faster foragers. Although females could benefit both directly and indirectly through their preference for faster foragers, the potential link between foraging efficiency and cognitive abilities was not investigated. 108. 109. Mountain chickadees. In this monogamous bird species, spatial cognition is important as mountain chickadees rely on the retrieval of previously stored food caches to nourish themselves during the winter. 110. Spatial memory has been shown to correlate with reproductive success in mountain chickadees. 111. When females mate with males possessing enhanced spatial cognition, they laid larger clutches and fledged larger broods when compared to females mating with males possessing worse spatial cognition. Therefore, when mating with high-quality males with enhanced spatial skills, females may attempt to gain indirect, genetic benefits by increasing their reproductive investment. 111. The Trinidadian guppy, Picilia reticulata, male above and female below. Other guppies. When assessing male cognitive ability via maze experiments, females preferred males that learn 2 mazes at a faster rate. 112. However, females did not directly observe males during their learning tasks. Although females could be discriminating between males by assessing orange saturation, a morphological trait weakly correlated to learning rate, females did not find males with brighter orange spots more attractive. Instead, females may use an unknown visual trait to discriminate between males and their cognitive abilities. 112. Meadow voles. In this species of rodents, female preference for male spatial ability was investigated. 113. Male spatial ability was measured using a series of water maze tests, before females were given a choice between 2 males. 113. 114. Although females did not directly observe males complete spatial ability tasks, they preferred males with enhanced spatial skills. This preference could explain why meadow vole brain regions associated with spatial ability are sexually dimorphic. Spatial ability could serve as an honest signal of male quality due to the metabolic costs associated with brain development. 98. 113. 115. Criticisms. Although there is some evidence that females assess male cognitive ability when choosing a mate, the effect that cognitive ability has on survival and mating preference remain unclear. 99. Many questions need to be answered to be able to better appreciate the implications that cognitive traits may have in mate choice. Some discrepancies also need to be resolved. For example, in 1996, Catchpole suggested that in songbirds, females preferred males with larger song repertoires. Learned song repertoire was correlated with the size of the high vocal center (HVC) in the brain. Females may then use song repertoire as an indicator of general cognitive ability. 116. However, a more recent study found learned song repertoire to be an unreliable signal of cognitive ability. Rather than a general cognitive ability, male songbirds were found to have specific cognitive abilities that did not positively associate. 117. 118. As of 2011, more research was needed on what extent cognitive abilities determine foraging success or courtship displays, what extent behavioral courtship displays rely on learning through practice and experience, what extent cognitive abilities affect survival and mating success, and what indicator traits could be used as a signal of cognitive ability. 99. Researchers have started to explore links between cognition and personality. Some personality traits such as boldness or neophobia may be used as indicators of cognitive ability, although more evidence is required to characterize personality-cognition relationships. 119. As of 2011, empirical evidence for the benefits, both direct and indirect, of choosing mates with enhanced cognition is weak. One possible research direction would be on the indirect benefits of mating with males with enhanced spatial cognition in mountain chickadees. 99. 111. Additional focus in research is needed on developmental and environmental effects on cognitive ability, as such factors have been shown to influence song learning and could therefore influence other cognitive traits. 99. See also: Extended female sexuality filter theory, sociology.